Hello and welcome to Gut Microbiome and Cardiovascular Health, Exploring the Connection. My name is Adair Anderson. I am the Product Marketing Educator here at Vibrant and want to go over some housekeeping items before we get started. All attendees are in listen-only mode. If you have questions during the presentation, please submit them as a comment. There will be time at the end for Q&A. Also, this webinar is being recorded and you will get a copy of the recording and the slides after the webinar ends. And now I'm pleased to introduce you to our speaker, Dr. Soheil Saeedi. Dr. Saeedi, PharmD, PhD, FESC, is a pharmacologist and scientist in the field of cardiovascular aging. He currently serves as a group leader and head of the Cardiovascular Aging Lab at the Center for Translational and Experimental Cardiology at the University of Zurich, and was formerly affiliated with the Cardiovascular Division at Harvard Medical School. Dr. Saeedi has received several academic honors, including the Paul Dudley White International Award from the American Heart Association, the Early Career Research Award from the International Society for of Thrombosis and Hemostasis, and the ADLA Walter Reason Award from the Swiss Atherosclerosis Association and the European Atherosclerosis Society Young Investigator Fellowships. He also received a postdoctoral fellowship at the Harvard Medical School in 2017. The Saeedi Lab investigates gut microbial metabolites and their regulation of redox signaling in orchestrating senescence in vascular endothelial cells in perivascular adipose tissue, and the subsequent cardiovascular aging. Dr. Saeedi believes this research, utilizing gut microbiome-based strategies, enables personalized and precision medicine for decelerating vascular aging in the growing elderly population. Take it away, Dr. Saeedi.
Thank you very much, Heather, for having me today. Uh, Hello, ladies and gentlemen. Welcome to our clinical webinar. Thank you for joining me today to discuss an intriguing and rapidly evolving area of research, means the interconnection between the gut microbiome and cardiovascular aging. Within the next, about for manipulating the gut microbiome for preventing or decelerating, uh, aging process in the cardiovascular system. And in the end, I will give you a few practical health advice to improve your gut health and decelerate aging process in the heart and vasculation. First of all, the critical question: what's the gut microbiome? 100 trillions of microbes live in your skin and inside your body. These communities of bacteria, viruses, and fungi make up your microbiome. However, when most people refer to their microbiome, they're talking about the many types of bacteria that live inside the gastrointestinal tract. This ecosystem, contains more genetic material than the human genome itself, and it plays a crucial role in maintaining our overall health. These bacteria perform several important jobs, including digestion of complex carbohydrates, proteins, and fat, and converting them, into absorbable nutrients. The second job is breaking down fibers and starch into some other, substances. For instance, when you eat an apple, your stomach and small intestine break down some of it. The rest of the apple goes to your colon, where bacteria do the rest of the work for you. As the bacteria break down the apple's fiber, they produce substances that are good for the cells of your colon and the rest of the body. These substances are so-called short-chain fatty acids that play essential role uh, in disease prevention, immunoregulation, and intestinal barrier integrity. Another job that the gut microbiome does is synthesizing essential vitamins and amino acids. For instance, vitamin B, B1, and vitamin K, which is essential for proper and normal coagulation in the body. Beyond digestion, the gut microbiome is instrumental in immune system regulation. Approximately 70% of immune system is housed in the gut, where it interacts with microbial communities to distinguish between harmful pathogens and beneficial organisms. It helps preventing infections and maintaining immune homeostasis. The production of short-chain fatty acids by the gut bacteria like acetate, propionate, butyrate, uh, has systemic anti-inflammatory uh, and effects, which are vital for maintaining the colon health and protecting against inflammatory diseases like inflammatory bowel disease or IBD, and also, preserving the intestinal barrier integrity. In general, the gut dysbiosis or imbalance in the gut microbiome has been linked to a variety of conditions, including obesity, diabetes, and cardiometabolic diseases. So, understanding these functions highlights the critical role of the gut microbiome as a major player in our health and sets the stage for exploring its connection to cardiovascular aging. So, now I'm going to give you some examples on the bacterial species which are associated with cardiovascular health and diseases. Emerging research shows that the gut microbiota profoundly influences cardiovascular health through its diverse bacterial community and the metabolites they produce. This body of findings has led to coining the term gut-heart axis, which has become increasingly recognized in the field. Here's a short list of beneficial bacteria known for their effects on cardiovascular system. For instance, we do have the Lactobacillus and Bifidobacterium species, which are famous probiotics that can produce different short-chain fatty acids like butyrate and propionate. And in addition to reactive peptides which have some effects like ACE inhibitors or angiotensin converting enzyme inhibitors, which are known medicines for lowering the blood pressure. They can also reduce the inflammation and preserve the lipid homeostasis. The third example is Acarmensium muciniphila, that's iconic, provided on in the field, and also is commercially available nowadays, and it's garnered attention for its role in cardiovascular health. This bacterium is known to enhance the integrity of the gut barrier and reduce inflammation. Clinical studies have found that the higher levels of this bacterium are associated with improved metabolic profiles and lower risk of cardiovascular diseases such as atherosclerosis and hypertension. Its beneficial effects are partly due to its role in producing short-chain fatty acid, particularly propionate, which can lower blood pressure and reduce cholesterol levels. They're also commercially available and used for lowering, the weight, and it's used for obesity, and also can control the blood glucose in patients with type 2 diabetes. Another example is Oxalibacter genius, which is very recently, a few minutes ago, was found to have mm, specific effects in breaking down the cholesterol and thereby reducing the accessible cholesterol in the circulation. So it could be used, as a supplement or adjunct therapy for, reducing the cholesterol levels in patients with familial hypercholesterolemia. Another notable bacterium is Bacterium prosnitzi, which produces butyrate, another, critical short-chain fatty acid that has anti-inflammatory effect. Reduced levels of this bacterium uh, and its byproduct, which is the butyrate, has been linked to increased systemic inflammation and higher incidence of cardiovascular diseases such as coronary artery disease and heart failure. Um, Other examples are there, and we can still talk about Prevotella, but this bacteria has some controversial effects. For instance, uh, in some studies, it's seen that the abundance of this bacteria increase during aging in the aged, gut microbiome. And in some cases, it's known as a probiotic that can somehow reduce the inflammation and could be beneficial for the cardiovascular system. Um, on the other hand, there's a long list of bad bacteria. For instance, Enterobacteriaceae species, which are often elevated, in aging and in gut dysbiosis and different diseases, have been associated with adverse cardiovascular outcomes due to their role in promoting inflammation and contributing to development of atherosclerosis. These findings underscore uh, the complex interplay between specific gut bacteria, their metabolites, and cardiovascular health, highlighting the potential for microbiome targeted therapies in managing cardiovascular diseases. Now it's time to move forward to the second section, which is uh, you know uh, the microbiome and cardiovascular aging. But before talking about the cardiovascular aging, I'm gonna shortly discuss, how gut microbiome and host affect each other during aging process. Since 16 years ago that Jeffrey Gordon and his team published a report in journal Science, it's increasingly known that many bacterial lineages have diversified alongside their mammalian hosts over millions of years. Today, we know that the microbiome and host simultaneously affect each other. It changes as the host ages, but it also modifies age-related impairment of the host and contributes to aging-associated diseases, including cardiovascular disease. As we age, the composition and function of our gut microbiome undergoes significant changes. These changes include a decline in beneficial commensal bacteria such as bifidobacterium, faecalibacterium, and lactobacillus, as I said, as famous probiotics, and increasing potentially pathogenic microorganisms or pathogens like proteobacteria, enterobacteria, clostridiacea, species. This imbalance, you know, is exacerbated by age-related changes in immune function and gut barrier integrity, leading to chronic low-grade steroid inflammation, so-called inflammation, which is a hallmark of aging. Inflammation can contribute to the development of cardiovascular diseases by promoting arterial stiffness, endothelial cell senescence, a hallmark of aging, and dysfunction, atherosclerosis, and atherosclerosis. Arterial stiffness is important because that's one of the hallmarks of vascular aging and has positive association with hypertension in old humans. Uh, Moreover, due to the dysbiosis, there is a reduction in the release of short-chain fatty acids, which poses anti-inflammatory properties, as I said. Consequently, disruptions, in key metabolic processes promote inflammation, oxidative stress, DNA damage, which are all the hallmarks of cellular senescence as another hallmark of aging process that induces tissue damage, tissue remodeling, and thereby contributes to pathogenesis and progression of age-related cardiovascular diseases. When you're born, your microbiomes only partially develop. For this reason, during the first few years of life, the microbiome is more vulnerable than during adulthood when the microbiome is more established. This is why repeated courses of antibiotic therapy of children, which is prolonged for months, will affect your microbiome more severely than one course of antibiotic taken for several days. Accordingly, the higher diversity of the gut microbiota and abundance of a specific microbial signatures like Prevotella, Bifidobacterium, Lactobacillus, Lactococcus in centenarians who are, the old healthy humans above 100 years old, is intriguingly associated with health span and longevity. So, in the definition of the reciprocal interconnection between gut microbiome and age, there are two theories. Of course, it's quite complex to say which one is the superior, But the first theory claims that the host aging influences the gut microbiome, developing abundance of harmful bacteria. And the second one, I'm very closer to this, states that alteration of gut bacteria and impaired diversity can promote aging process in the host system. For each of the theories, there are different evidences. For instance, for the first theory, based on our and others' studies, the gut microbiota undergoes dynamic changes during host aging. Changes in the host intestinal cell composition and architecture occurring during aging are matched by a decrease in the microbiota diversity that leads to enrichment of new age-associated microbial species with potential of pathogenesis. For instance, on the right side, on the top, you can see that there's a decrease in alpha diversity, and at the bottom, you know, the differential beta diversity in the gut microbiome of old versus young mice. And this comes from our previous study, and it shows clearly that during the aging process uh, in the host, we do have changes in diversity, even in its richness and uniqueness of the gut microbiome. Another example is in progeria mice. That's a mouse model of premature aging, and the animals mimic the Hutchinson-Gilford progeria syndrome or HGPS. And still in the premature aged mice, we can see the gut dysbiosis and alteration of the gut microbiome, which makes them, more similar to normal aged microbiome. So it shows that these evidences support the first theory that host aging can promote the gut dysbiosis and alteration of their composition. The second theory is supported by other evidences. For instance, the first one, uh, you know, there are some studies in the literature, but I'm showing the data from our own studies in my own lab. And when we did the fecal microbiota transplantation or FMT from old to young mice, we observed, that these young mice mimic the aging phenotype in their cardiovascular system. And also, when we did the FMT from young mice to old mice, interestingly, we observed that, there is a deceleration in the aging process in cellular senescence in aortic endothelial cells and perivascular adipose tissue of old mice. And it was accompanied by remarkable changes in the composition and diversity of uh, the gut microbiome in old mice that receive FMT from young mice. And in this wind plot, you can easily see that uh, the gut microbiome of old mice that receive you know the FMT from young ones is similar to what we see in the normal young mice. Other evidences, come from the contribution of the bad microbes and metabolites to unhealthy aging. There's always a fight between, good and bad bacteria and metabolites. Of course, the picture, that's the fight between, you know, the good and ugly, but good and bad. For instance, when we have the gut dysbiosis and higher abundance of disease-associated bacteria, they can promote the aging phenotype in diverse tissues of the host. For instance, the lower ratio of the firmicutes to bacteroidota may be associated with cardiovascular diseases. This ratio is also, measured, within the gut Zoomer sheets and tests of the Vibrant Wellness company, and it can be one of the markers that shows that the gut microbiota is aged or still young, and that could be a parameter for microbial age metric. Uh, Anyway, the lower ratio of the firmicutes bacteroidota is associated with cardiovascular diseases, including higher thrombotic risk, heart failure, uh, atherosclerosis, et cetera, to the extent seen in aged humans and animals. At family level, increases in Staphylococcacia, Klebsiella, Clostridioideaceae, and Bacteroideaceae are associated with coronary artery disease, heart failure, ischemic heart disease, so on, and et cetera, et cetera. And the enterotypes of large aging cohorts from Japan, Italy, Greece, reveal that, the gut microbiome of centenarians has features that are usually associated with gut microbiomes of young individuals. It means that they do have increases in, microbial species evenness, uh, richness, and enrichment of potentially beneficial bacteria, this Prevotella and Lactobacillus bifidobacterium, and depletion of potential pathobionts. So these are other, evidences that support the second theory. But anyway, the secret to successful aging, lies in part in our gut. So it may be possible to predict our likelihood of living a long and healthy life by analyzing the trillions of bacteria, viruses, fungi that inhabit in our intestinal tract. And the question is that what's the chicken and what's the egg? Host aging regulates gut microbiome aging, or aging microbiome induces host aging. Uh, Altogether, what's important is the positive association between gut dysbiosis or alterations and aging associated diseases, including especially cardiovascular disease like vascular dysfunction, atherosclerosis, coronary artery disease, atrial fibrillation, heart failure, hypertension, thrombotic risk, and cardiovascular mortality. Um, I just want to give a few sentences, about the cardiovascular disease in aging and its importance, because we all know that, still the number one killer of the people around the world is cardiovascular diseases, and it takes about, 18 millions of lives every year around the world, more than, uh, what different types of cancer do. So it's really important to note its underlying mechanisms, what happens during aging that makes the people more prone to cardiovascular diseases, what's the role of the gut microbiome, and how we can explore new strategies for preventing or decelerating the aging process in the cardiovascular system to reduce the number of these deaths. Uh, Based on a very recent paper published in Nature Medicine, uh, the gut microbial age, I already indicated in previous slides, gut microbial age modulates cardiovascular disease risk in metabolically unhealthy older people. It states that patients with higher microbial age are more positively associated with cardiovascular diseases. In their microbiome, diversity has dropped, and short chain fatty acid producing bacteria are less abundant, while Enterobacteriaceae and Prevotella became more abundant. You remember I said that controversial effects of Prevotella. And what's again seen is the correlation of the microbial age and host age. Therefore, microbial age could be potentially considered as a clinical parameter for protection of cardiovascular diseases in long term. In this picture, you can see that, there's a confirmation for their findings that shows a decrease in gut microbiota richness and butyrate, short chain fatty acid, producing bacteria and expansion of pathobionts in patients with cardiometabolic and cardiovascular diseases that demonstrate BCD, resistance, myocardial infarction, or heart failure. Okay, so let's talk about uh, the metabolites of this gut bacteria and their connection to cardiovascular aging. We discussed, the positive association of some specific bacteria with cardiovascular aging process. What's important now is their products, which are the metabolites. We know that, the gut microbiota produces thousands of chemical products, so-called metabolites, from blind acids to short-chain fatty acids that act as signaling molecules to regulate diverse metabolic processes and actively control healthy aging. During the recent 2 decades, it's more understood that the alterations of gut microbiota and its metabolites play a crucial role in the pathogenesis of aging-associated diseases and cardiovascular disease. The gut microbiota, undergoes extensive alterations with age and is seen in all patients with cardiovascular diseases, particularly atherosclerosis. For instance, with advanced age, gut microbiota alterations might contribute to, uh, you know, cardiovascular disease through extended, oxidative stress and inflammatory responses. And one such example is the accumulation of uremic toxins, which produced from aromatic amino acids, phenylalanine, tyrosine, or tryptophan, that lead to accelerated aging of the cardiovascular system. For instance, phenylacetylglutamine, which is metabolized from the phenylalanine, is positively associated with the development of atherosclerotic plaques, coronary artery disease, and major adverse cardiovascular events, or MACE, in diabetic and non-diabetic patients. Uh, and recently, from the Stanley Hazen's group in Cleveland Clinic, they also showed that there's a positive correlation between the higher plasma concentration of and this metabolite with heart failure as well. Another example is trimethylamine inoxide, or TMAO. We and others, have demonstrated that TMAO contributes to the pathogenesis of a wide range of cardiovascular diseases by promoting endothelial cell senescence, dysfunction, vascular dysfunction, thrombosis, and plaque formation. So uh, we know that you know two major series of metabolites, TMAO and metabolites of aromatic amino acids, are nowadays known for their causal and mechanistic effects on cardiovascular diseases during aging. The first group is metabolites of aromatic amino acids. The gut microbiota significantly influences the metabolism of, these aromatic amino acids, which play crucial roles in cardiovascular health, as I said. Gut microbiota can metabolize these amino acids into various bioactive metabolites, impacting systemic inflammation and cardiovascular function. For instance, phenylalanine is converted by the gut microbiota into phenylpropanic acid, and then phenylacetic acid, and finally phenylacetylglutamine, that can influence blood pressure and endothelial function and senescence. High levels of phenylalanine and its metabolites have been associated with increased arterial stiffness and elevated risk of cardiovascular mortality, highlighting its impact on cardiovascular health during aging. Tryptophan, another aromatic amino acid, is converted by gut microbiota into several metabolites, including indoles and its derivatives, which have been shown to modulate immune response and inflammation. Disrupted balance in tryptophan metabolism can lead to increased levels of inflammatory cytokines and atherosclerotic changes in the vasculature. Diet plays a critical role in shaping this microbial metabolism. A diet high in proteins, particularly those rich in aromatic amino acids, can affect microbial composition and metabolism, and potentially, lead to adverse cardiovascular outcomes. And we know that these aromatic amino acids are mostly found in Western diets. In next slides, I will talk about the different diet, uh, including Western diet and Mediterranean diet. Um, Conversely, dietary fibers and polyphenols can influence, microbial diversity and the production of beneficial metabolites, potentially mitigating cardiovascular risk. For instance, eating stress plants rich in polyphenols, like green tea, leafy greens like spinach, can activate the body's stress responses. Therefore, the interplay between diet, gut microbiota, and aromatic amino acids metabolism is crucial in managing cardiovascular aging. Here, in the summary of these results from a study conducted by our colleagues in Germany, Switzerland, United States, And you can easily see the positive correlation between the plasma concentrations of these aromatic amino acids with cardiovascular mortality and MACE. So this confirm what people, including us, observe that in vitro and in vivo animal models. Among these three aromatic amino acids, in my own lab, I mainly focused on phenylalanine. And we know that phenylalanine is enriched in, you know, red meat, processed food, dairy products, cheese, or French fries, something like that. In addition to, energy drinks and the zero soft drinks that contain aspartame, cyclamen, sodium saccharin, and you can easily see that you know the presence of phenylalanine in the chemical structure of the aspartame. We know that the phenylalanine in this food are metabolized by the gut bacteria harboring more PP4 or PPDC gene homologs, and they metabolize phenylalanine into phenylacetic acid or PAA. And then PAA is converted to phenylacetylglutamine in the host liver. To see whether, these metabolites have causally effects on uh, the vascular aging in both human and animal models, we first focused on, the samples that we collected from the patients with acute coronary syndrome from enrolled in the spumaceous cohort. That's a Swiss-based multicentral cohort in about 4,800 patients. More than 90% of them are old, about 65 years old, and in within the patients in the first part of this cohort, in about 1,800 patients, we observed the higher concentration of these two metabolites, PAA and PAG. And they were positively associated with MACE, 1 year MACE and mortality. We did, we continued the studies in normal aging model. And we collaborated with the Twins UK cohort department in King's College London. And again, in more than 7,300 healthy humans from the age of 18 to 95, we observed a positive correlation between the plasma concentrations of both PAA and PAG. Here in this slide, I only show the data for PAA with age. And in 900, subjects out of this, we observed the positive correlation between the abundance of Clostridium genius with plasma concentration of PAA. We also did, the same study, the same, subset of studies in normal aging model in mice, and observed that only one bacterium, which is the Clostridium ASF356, is positively associated with plasma PAA concentrations. So we knew that PAA is associated with cardiovascular mortality in old humans. And we also observed that, only one specific bacterium, at least in animal model, and it was extrapolated to humans, as you can see in the middle, was responsible for generation of PAA. So we thought that whether, this metabolite can causally induce the cellular senescence as hallmarks of aging in the vasculature. So we did the in vitro and in vivo studies and observed that, the PAA per se can induce the cellular senescence in aortic endothelial cells and also in perivascular adipose tissue. As a proof of the concept, we did the colonization of young mice, this bacterium, and then we observed that, the aortic endothelial cells recapitulate almost all the hallmarks of cellular senescence. In few sentences, I will tell you what the senescence is. The cellular senescence uh, is the replicative arrest in the cells, and the cells become dysfunctional by increasing the hallmarks of cellular senescence, including oxidative stress, mitochondrial dysfunction, epigenetic alterations, the upregulation of senescence-associated secretory phenotype or SASP components, including pro-inflammatory cytokines, chemokines, adhesion molecules, um, DNA damage response, and telomere shortening. And finally, the cells don't proliferate anymore and they're dysfunctional, as we said. And they can secrete this, senescence promoting secretin towards the adjunct cells. And they can induce the cellular senescence in neighboring cells. It's why, they're called zombie cells because they're dysfunctional, they don't die, and they can, spread, uh, this disease means, this pathological state in the neighboring cells. For instance, we observed that, aortic endothelial cells in the colonized mice show the upregulated interleukin 6 as a SASP component and DNA damage response represented by gamma H2AX uh, plus the upregulation of cell cycle arrest marker, which is the P16 in the cell cycle of the endothelial cells, accompanied by increased upregulation of another SASP component, which is the VCAM1, which is also a biomarker for endothelial cell dysfunction and atherosclerosis. Okay, let's talk about another important metabolite that can contribute to cardiovascular aging. That's TMAO or trimethylamine oxide. It's derived from the gut microbiota's metabolism of dietary choline, phosphatidylcholine, and L-carnitine. Diet plays a crucial role in the production of TMAO, as it's primarily, derived from the metabolism of dietary nutrients, which are abundant in food like, red meat, eggs, dairy products, processed food. Again, you see the Western diet. Consuming a diet rich in these nutrients can increase TMA levels. It's been gained, significant attention as both a biomarker and contributing factor in cardiovascular diseases. Elevated levels of TMAO in the blood, have been associated with increased risk of cardiovascular events, including heart attack, stroke, and atrial fibrillation. Mechanistically, TMU is thought to contribute to cardiovascular disease through several pathways, including, promoting cholesterol deposition in arterial walls, enhancing platelet hyperreactivity and thrombotic risk, and contributing to inflammation and oxidative stress. These processes can accelerate the atherosclerosis and increase the risk for thrombotic events. We also, in both human and animal studies, observe how this metabolites is associated with cardiovascular diseases during aging. For instance, in one clinical cohort, again Swiss-based and multi-central, which is called the Swiss AF, in about 2,400 patients, old patients, older, about 65 years old, all patients with atrial fibrillation that have the history of stroke. We observed that there is a positive association between the third tertile of plasma TMAO levels, cardiovascular mortality, and also, as you can consider here in the lower panels, there's a positive correlation between, TMAO levels and mortality, uh, stroke size, and injury size. In animal model also, we could see, uh, some harmful effects of TMAO. For instance, here in this heat map that comes from the 16S RNA sequencing of old and young, micro, microbiome from mice, you can see that there is a, remarkable difference between the composition of the gut bacteria. And it was accompanied by increase in the abundance of, TMA, the precursor of the TMAO, producing TMA producing bacteria like the sulfovibrio, that increased the plasma concentration of TMA. And it was associated with increased pro-inflammatory responses, especially the SASP components, interleukin 1 beta and 6. And uh, also, the TMAO could increase the hyperresponsive the platelet hyperresponsiveness to stimulants and thrombotic risk. And in this take-home figure, you can see the summary of these findings, which was published 3 years ago in iScience. And we could see that aging is associated with increased in the abundance of TMA-producing bacteria that increases the inflammation, oxidative stress, platelet hyperresponsiveness, and thrombotic risk. Okay, now it's time to move through the fourth section, which is the connection of the diet, gut microbiome, and cardiovascular health. As earlier indicated today, diet potentially modulates the gut microbiota by regulating the balance between pathogenic and beneficial microbes or microbial products, metabolites. Vegetarian diets foster a beneficial microbiota composition by increasing Prevotella, uh, for instance, or Lactobacillus lactococcus, bifidobacterium enterotypes, whereas diets high in animal protein foster Bacteroides, Klebsiella, Bilophila enterotypes, and other species associated with proatherogenic metabolites and cardiovascular disease. For instance, the production of the proatherogenic metabolite TMAO, resulting from TMA oxidation by FMO3 enzyme in the host liver, and its release into the systemic circulation are linked to coronary plaques, peripheral artery disease, and severe cardiovascular disease and its complication, including stroke, myocardial infarction, and death, as I shortly indicated in previous slides. Common dietary nutrients that contain TMA precursors, such as choline, phosphatidylcholine, L-carnitine, present in red meat, fish, dairy products, egg, and these are the main contributors to TMA production and, make the patients more prone to atherosclerosis. These are examples. So it's quite important what we eat. We have Western diet on one hand, and we have Mediterranean diet on the other. And of course, there are some other, diets, but I'm going to talk about, the extremes, uh, Western and Mediterranean. So the Western diet characterized, by high intake of red meat, processed food, saturated fats, and refined sugars, and their consumption is increasingly associated with the prevalence of cardiovascular diseases in aging populations. For instance, in uh, this panel, you can see, during the recent 2 decades, especially, there is a strikingly increase in the consumption of red meat and processed food. Um, on the globe. So, in addition, this diet often leads to elevated levels of aromatic amino acids like phenylalanine and tyrosine, key contributors to cardiovascular disease, as already indicated. In addition, red meat and processed food are particularly concerning, due to their high levels of saturated fats and preservatives, which can elevate cholesterol levels and promote atherosclerosis. As people age, the cumulative effects of such dietary patterns can lead to higher incidence of heart disease, stroke, and other cardiovascular conditions, underscoring the importance of uh, you know dietary modifications for healthy aging. For instance, here you can see, based on a study, uh, there is a positive association between all-cause mortality, cardiovascular mortality in both sex with red meat intake. And also in another study, you can see that, for instance, phenylalanine is positively associated, plasma concentration of phenylalanine is positively associated with cardiovascular and cerebrovascular events like, stroke. On the other hand, we do have the Mediterranean diet. We know that the Mediterranean diet, rich in fruits, vegetables, whole grain, nuts, and olive oil, is renowned for its benefits to cardiovascular health. Recent research, highlights its role in modulating the gut microbiome, which plays a crucial part in the protective effect. Uh, the diet increases, the abundance of beneficial gut bacteria like lactobacillus, lactococcus, and bifidobacterium, known probiotics, which are linked to anti-inflammatory effects and improved lipid metabolism. It promotes the production of short-chain fatty acids and other metabolites such as polyphenols, which are known to reduce the hallmarks of cellular senescence, including cellular stress, DNA damage, and SASP, upregulation of the SASP components. Both key factors, all key factors in cardiovascular diseases. Our and others' studies demonstrated that the secretion of short-chain fatty acids, particularly acetate, which negatively associated with inflammation and cardiovascular diseases, including atherosclerosis and blood pressure, reduces with age and causes cardiovascular complications. You see that on the left side that during in old mice we do have the significantly lower concentration of both acetate and propionate as short-chain fatty acids in both fecal and plasma samples, and these are accompanied by upregulation of thrombotic factor like tissue factor and pi1 that subsequently results in increased thrombus formation and thrombotic risk. Uh, you know, uh, in addition to this, in human studies, it's demonstrated that the secretion of short-chain fatty acids, uh, you know, can somehow be associated with the lower risk for cardiovascular diseases, cerebrovascular diseases. And there are so many nowadays, and the literature is getting uh, overwhelmed with the findings on the effects of the short-chain fatty acids on the prevention or improving the cardiovascular health in the old organisms in both mouse and human models. Taken together, the balance between the production of these positive or good and negative or bad metabolites may regulate the vascular aging, cardiovascular aging process and cardiovascular function. So their balance are important, of course. So let's talk about the new strategies for preventing or decelerating the aging process in the cardiovascular system. There's a slogan: if we want to die when we're old, so first of all, we have to not to die uh, when we are young. Okay, so what should we do to stay young? One strategy is using uh, prebiotics or probiotics or postbiotics or symbiotics. What are prebiotics? Uh, Prebiotics may feed certain beneficial colonies of gut bacteria. Prebiotics found in food, like garlic, onions, bananas, stimulate, the growth of beneficial bacteria and enhance the production of short-chain fatty acids. Polyphenols present in food, like, berries, nuts, green tea, also support the gut health by promoting, the growth of beneficial bacteria and exerting, anti-inflammatory effects. In clinical trials, prebiotics such as inulin and fructooligosaccharides have been found to enhance the gut microbiome diversity and reduce risk factors associated with cardiovascular diseases, including lowering blood pressure and improving lipid profiles. There is an ongoing trial registered in clinicaltrials.gov is exploring the effects of prebiotics fiber uh, supplementation on cardiovascular health outcomes. But still, there are very few clinical trials working on, prebiotic, probiotics or, postbiotics. But, the clinical trials are getting expanded during the recent years. Another strategy is intake of probiotics, which are live beneficial bacteria found in fermented food like yogurt, kefir, that can help restore and maintain the healthy microbiome. Evidence suggests that probiotics can improve lipid profiles and reduce inflammation, which are critical for cardiovascular health. Clinical trials also, have demonstrated that probiotics can reduce levels of LDL cholesterol and inflammatory markers. I guess in the states, there are only 6 or 7 clinical trials ongoing or recruiting working on the probiotics, since probiotics are still have some challenges and that their effects are not durable or reliable. You know, at the level to be used as a treatment. Still, they can be used as adjunct therapy. Similarly, postbiotics, which are the metabolites of the bacteria, which are produced by probiotics, are being explored for their cardiovascular effects. Postbiotics like short-chain fatty acids have been shown to lower blood pressure, reduce arterial stiffness in clinical settings. Especially butyrate products are used and are very limitedly, commercially available nowadays. Symbiotics, which combine probiotics and prebiotics, aim to enhance the effectiveness of both therapies. And these are, there are clinical trials evaluating the impact of the symbiotic supplementation on metabolic syndrome and associated cardiovascular risk. I guess only one ongoing clinical trial exists, on clinicaltrial.gov in the States, and they're working on, uh, you know, the effects of the symbiotics on blood pressure. As an example, in the case of the prebiotics, in my own lab, in collaboration with other researchers in our university, we also examine the effects of the probiotic omega-3 fatty acid, alpha-linolenic acid, which is supplied from herbal sources like flaxseed oil or walnut oil. We supplemented for long term and supplemented old mice for long term with this probiotic, and we observed the remarkable, changes in the composition of the gut microbiome. It was associated with the shift from inflammatory, promoting and harmful bacteria towards the short chain fatty acid producing and health associated bacteria. And it was also accompanied by increase in the concentrations of acetate and propine in the short chain fatty acids in both fecal and plasma samples that reduce the inflammatory responses, especially by reducing the SASP components, interleukin 6 and interleukin 1 beta, that finally could reduce the thrombus formation and thrombotic risk. We also, tested how the supplementation with this probiotic for long term could improve the cardiac health. So we observed that these probiotics could improve the diastolic function of old mice and could also prevent the tissue remodeling and the fibrotic area in the heart of the old mice. In the case of the postbiotics, we also, based on the findings that showed that there's a decrease in plasma and fecal concentration of acetate, we supplemented aortic endothelial cells from old mice with sodium acetate, and interestingly observed that this postbiotic can rescue the cellular senescence in these cells. For instance, we could see that there is a reduction in SA beta gal activity and increasing the proliferative marker expression KS67, and accompanied by a reduction in gamma H2AX as a marker for DNA damage response and increasing you know, telomere length presented by increasing expression of the human TERT, velocity reduction in SAS components, interleukin-1 beta and 6, and oxidative stress. And all these finally improved the mitochondrial respiration and improve the function of filial cells by increasing the filial cell sprouting as marker for angiogenesis capacity. So this postbiotic could somehow rescue cellular senescence as a hallmark of aging in aortic and cells. What are the future uh, strategies for manipulating gut microbiome to decelerate the aging process in the host cardiovascular system? And medical interventions can play a significant role in managing gut microbiome health, particularly in addressing these biases and its implications for cardiovascular health. One promising intervention is fecal microbiota transplantation, or FMT, as I already indicated in previous slides. FMT involves the transfer of a stool from healthy donor to a patient with microbiome imbalances. This procedure has shown a potential in restoring the microbial diversity, improving metabolic and inflammatory profiles. FDA has approved two microbiome-based therapies for the prevention of the recurrence of Clostridium difficile infection, which is common in older individuals, especially those hospitalized. And fecal microbiota transplantation uh, is one of them. And the fecal microbiota products also, show. Also show the promise for other diseases associated with gut dysbiosis like inflammatory bowel disease or IBD and metabolic syndromes. Despite this milestone, challenges remain, including the need for long-term safety data, potential risk of pathogen transmission, and ethical considerations. Advances in microbiome sequencing by 6ns RNA sequencing and shotgun metagenomics and bioinformatic analysis are paving the way for personalized microbiome editing that could optimize cardiovascular health on a case-by-case basis. The goal of bacterial genetic engineering and microbiome editing uh, are a precise edition of the genomes of the bacteria associated with different diseases in humans. The CRISPR machinery and base editor approaches, uh, carried with engineered delivery vehicle using components of bacteriophages, can specifically, remove the bacteria's capacity to produce the pathogenic molecules and the bad metabolites while retaining its other functions. However, there's a long road that, ahead and challenges remain. One is that, the approach uses short-lived delivery of gene editing machinery to make gene disruptions. In addition, identifying, deliverable phages, bacteriophages, is another challenge. Of course, it's a kit. So, and also not all targets have been, discovered to edit. Uh, in my own lab, we also utilized the bacterial genetic engineering approach based on our findings on phenylalanine derived metabolite phenylacetic acid. You remember that I told you that the gut bacteria harboring WAR and PP4 and PPDC gene homologs can metabolize phenylalanine towards phenylacetic acid. And Clostridium ASF356 was positively associated with plasma concentrations of PA. So, based on these data and others, from the Stanley Hazen's group, that they showed that there's a positive association between the abundance of bacteria harboring PP4 with atherosclerotic cardiovascular disease, we decided to do the genetic engineering of this bacteria, and we depleted the PP4 gene, which was responsible for metabolism of phenylalanine to phenylacetic acid, and then colonized mice with this mutant bacteria. And we interestingly observed that the cellular senescence hallmarks, including the P16 and also the gamma H2AX, which was a marker for DNA damage response, were significantly lower in the mice colonized with wild type bacteria. And the mutant colonized bacteria also showed a higher angiogenesis capacity with with higher number of the endothelial sprouts, as you can see on the right side, plus a higher vasorelaxation vascular function responses compared to wild type colonized mice. So, that was exactly, the marriage between, bacterial genetic engineering and also, uh, you know, the uh, understanding of the mechanisms of the bacteria for cardiovascular aging. Um, Now it's the time to shortly, hint the importance of the lab test for prediction of the cardiovascular senescence and aging for preventing, the cardiovascular diseases during aging. Uh, we are now collaborating with, wider wellness company, and we're working on the samples from a Swiss-based, you know, central cohort, which is called aging heart survey cohort. And in this cohort, we do have, about now, today, I can tell you that about, 1,000 patients with old patients, all are above 80 years old, with four different cardiovascular diseases: heart failure, atrial fibrillation, arrhythmia, and coronary artery disease. And we collected plasma, urine, and stool samples. And we are working on the first part of this cohort with 105 patients. And we are doing the gut tumor, test for, you know, uh, having a comprehensive picture about the gut microbiota diversity and composition in these patients. I believe that, the gut zoomer will give us a picture of the gut microbial diversity and composition, and can also, provide a predictive, metric for cardiovascular disease as the host ages. Like you remember, I said that in the microbial age from the Nature Medicine paper, uh, you know, uh, that's about the microbial age. And the gut zoomer has the potential to be used as a microbial age metric based on the age associated microbes. In addition to this, and we plan to test the cardiovascular profile, especially, the lipid profile, as well as the methylation profiling of these patients, because that would be really important for the longevity and, the health plan. And it could be used, for mm, doing the studies for association between, the gut microbiome composition and epigenetic clock. Plus, we plan to run oxidative stress profiling, and especially for instance, OHDG biomarker is really important for cellular and DNA damage, which is one of the hallmarks of cellular senescence. Another purpose of this study is finding the causal connection of the specific bacteria. For instance, like Bacteria theta for generation of the harmful ceramides with the prevascular adipose tissue and aortic endothelial cell senescence and aging. The study is still ongoing, and next week, I guess, we will have a meeting to discuss the first round of the readouts and results. So now I do have, some advices for you. Uh, First, be careful about your diet. Eat a wide variety of fruits, vegetables, beans, whole grains, and eat estrus plants and non-saturated oils like alpha-linolenic acid. Uh, Eat fermented, food like yogurt, kefir, less protein. Uh, Actually, improves the health markers, and limit your alcohol intake. A Mediterranean diet can potentially guarantee the gut health. Take a look at the Sardinians. Another advice is about the lifestyle. Adopting certain lifestyle changes can significantly enhance the gut microbiome health and consequently cardiovascular health. Regular physical exercise is one, such change that's been, shown to positively influence the gut microbiome. Exercise promotes microbial diversity and increases the abundance of beneficial gut bacteria, which in turn can reduce systemic inflammation and improve metabolic health. For instance, researchers indicate that physical activity can lead to an increase in beneficial bacterial species such as Carmensium muciniphila, which is associated with improved metabolic health, lowering blood glucose, and lowering weight. Additionally, effective stress management is crucial for maintaining a healthy gut microbiome. Chronic stress can disrupt the balance of the gut bacteria, uh, leading to gut dysbiosis and subsequent inflammation. Techniques such as mindfulness, meditation, yoga, and adequate, sleep have been shown to positively affect gut health by reducing stress-related disruptions. For instance, studies have demonstrated that stress reduction interventions can restore microbial balance and lower levels of cortisol. And here you can find, the routes for contacting me on X, LinkedIn, and you can contact me via my email. Thank you very much for your time and attention.
Okay. Thank you so much for sharing your research on the connections between the diet microbiome and cardiovascular health. Particularly, I loved how phenylalanine from red meat and zero-calorie sweeteners can turn into PAA, which increases the cellular senescence, and also how gut microbes like disulfide Vibrio can convert choline and carnitine into disease-promoting TMAO. And then also how you highlighted the chicken versus egg situation where aging can negatively impact our microbial diversity, just as gut dysbiosis can also accelerate aging and cardiovascular health risk. And finally, how you finish it up with things we can do to improve our gut microbiome health with our diet and our lifestyle. Let's open it up for questions now. If you have questions, please enter them in the comment section and we will put them on the screen as they arise. We can answer them live. So while we're waiting, I had one question, which is if phenylalanine increases PAA, and people who have the genetic condition PKU can't have as much phenylalanine, does that mean those people who avoid phenylalanine have decreased risk of cardiovascular disease? Or because they can't metabolize it, they actually have increased risk because it stays in their system longer? Do you know anything about that?
That's really great question. In general, the patients with phenylketonuria, especially during adulthood, suffer from vascular dysfunction and arterial stiffness, and they're more prone to atherogenesis. So, of course, there is a lack of the phenylalanine hydroxylase (PAH), but you know that's not the only reason for suffering such and such vascular diseases. Of course, the accumulation of PAA phenyl acetate is one of the major reasons behind the increased risk for vascular diseases. Still, it's not very well studied. I know that in the literature, I try to find some clue of the association between the increased PA concentration and vascular disease in these patients. But of course, there's accumulation. So we do have the PAA, and that's exactly one of the you know things we're working on that in our ongoing project, and we are working on collecting some samples from the PKU patients to see whether the accumulation of the PAA is the causal contributor to vascular disease in patients. Yes.
Wow. Thank you for that detailed answer. I can tell you're a scientist. You really dive in. All right. Next question from Tarana. How the gut microbiome studies in mouse models could be extrapolated to humans? Is that possible?
Uh, Great question. Good question for a basic scientist, of course, basic interstitial scientist. But uh, in general, less than 3%. In one study, it says that 2.65% of bacterial species, are shared between mice and humans. But over 80%, in some studies, it says 89 to 90% of notable functions are shared. So it means that, for instance, if we pick a specific bacterium from the microbiome of a human, we might not find exactly that bacterium in the mouse gut microbiome, but we can find the gene homologs in that bacteria. It means that, for instance, in the case of what I already presented, we have the Clostridium ASF356. We couldn't find it in humans, but we could find another specific bacterium that has more than 90% share a notable function and gene homologs. So it means that, yes, we can extrapolate our findings in mice to humans because we have about 90% shared and notable function.
Oh, that
Yeah,
makes so much sense. It's not just about the microbes, it's about what the microbes do, their function. Like
so
there's some microbes that produce butyrate, some microbes that produce propionate, and as long as you have those, you get the effect.
exactly.
All right.
Yeah,
Thank you for that.
my pleasure.
All right. I'm not seeing any other questions in the chat, and I know it is quite late your time in Zurich. So I'm going to say thank you, everyone, for joining, and I hope you have a vibrant rest of your day. Take care.
Thank you very much.